Professor Jane Rudolph's Microbiology Final Exam ARQ number 3, APC and Antigen Presentation. 1. Phagocytosis or endocytosis of antigen into the antigen-presenting cell, macrophage for example. 2. Partial digestion of antigen into peptide fragments in the phagolysosome. 3. Vesicles containing peptide fragments and MHC-II molecules merge, vesicle coming from Golgi. 4. Peptide fragments bind to MHC-II molecules. 5. Vesicle fuses with plasma membrane. Each will bind to different T cell receptor on different helper T cells. 6. Professional antigen-presenting cells present exogenous antigens in association with MHC-II molecules. The key indicates that rectangular, triangular, and square shapes represent antigenic peptide fragments, and that the notch in the MHC-II represents the CD4 binding site. Here is what Professor Rudolph's diagram looks like. To help our memories, let's go through it a little slower this time. ARQ number 3, antigen presenting cell and antigen presentation. Step 1 is phagocytosis or endocytosis of the antigen, which is drawn as a loopy line with a rectangle, triangle, and square attached to it, and it is phagocytized into the antigen presenting cell or macrophage, for example. Step 2 is the partial digestion of the antigen into peptide fragments. Make sure you label the phagolysosome. In step 3, vesicles containing peptide fragments and vesicles containing MHC-II molecules merge. The vesicle containing MHC-II molecules is coming from the Golgi apparatus, which I forgot to label in my illustration, but please don't forget to label in your ARQ. In step 4, peptide fragments bind to the MHC-II molecules from those merged vesicles. And in step 5, the vesicle fuses with the plasma membrane, allowing the peptide fragments bound to the MHC-II molecules to be displayed on the cell's surface. Now, because each of these antigens is different, each will bind to a different T cell receptor on a different helper T cell. Step 6 is simply a note that professional antigen-presenting cells present exogenous antigens in association with MHC-II molecules. So any antigen coming from outside the cell needs an MHC-II molecule to be presented. Don't forget to draw the key as well. In the key, you need to indicate that the square shape, the rectangle shape, and the triangle shape all represent antigenic peptide fragments. You also need to draw the MHC-II molecule again, and then point out the little notch in the side represents the CD4 binding site. And that's it for ARQ number 3.